Hello everybody, this is my evolution presentation and the paper that I'm researching is over the sexual selection and the adaptive evolution of the PKDREJ protein in primates and rodents. So PKDREJ is a testis-specific protein thought to be located on the sperm head surface. Specifically, it stands for, in humans at least, polycystic kidney disease and receptor for egg jelly related protein precursor, which it has a correlation with something else we'll talk about a little bit. That's just for in humans. In other species, it's just basically the name of the part of the gene that codes for the protein. That's kind of what it refers to. These types of proteins are highly conserved, meaning that negative mutations are removed rapidly due to decreasing the fitness of the sperm itself. So next topic that leads into is sperm competition, also known in this paper commonly as post-copulatory sexual selection, meaning it's a type of selection after the sexual event or sex in whatever species it is or reproduction event has already occurred. Sperm competition in a polyandrous system creates a specific environment for favoring sperm quality, quantity, swimming ability, and binding ability, and a couple other things as well. So polyandrous specifically just refers to females that mate with more than one male during a single breeding season. So basically it could be in a single mating event or something like in the same day or something like that, just when in this context at least, creating a specific scenario for sperm competition, so where there's multiple sperm competing for the same egg. The researchers wanted to find out if the PKDREJ protein was a target of post-copulatory sexual selection, or a target of sperm competition. Because proteins involved in reproductive processes show accelerated evolution, the rapid divergence of this protein indicates positive selection driven by sexual selection. The researchers in this case use primates and rodents due to their wide variety of mating strategies, which increases the likelihood for a polyandrous system, like we discussed earlier, to occur. So even though that they do mate in a wide variety of strategies, it's not always going to be prevalent that they're going to be mixing sperm with a single egg in presence. So the sperm competition doesn't always take account, but in these specific species that they chose to use in the study, their mating strategies are much more variable than most others, so that's why they use these. So the researchers didn't state a specific hypothesis. They talked about it a little bit in their introduction, as far as you can kind of tell what they were hoping to accomplish and hoping to find from the study, and we'll talk about a little bit if they did or not. But the purpose of the study, which is kind of similar to the hypothesis, is to test for positive selection in the PKDREJ of primates and rodents and examine whether within these two groups separately, lineages experiencing high levels of sperm competition show a higher evolutionary rate of PKDREJ. And we'll go over how they measured both of those things, be it sperm competition and the evolutionary rate of PKDREJ. So the researchers used 12 primates species, and the PKDREJ sequences were actually obtained from the species because they were used in a previous study. So they didn't have to do any specific techniques for the primate species to obtain their DNA, because they were used from previous studies. And one phylogenetic sequence was built for the evolution of PKDREJ, and a second analysis aligned the REJ domain to compare the evolution of this region to the regions, how it compared to on rodents. So the REJ we didn't mention earlier, but in both primates and in rodents, they focused as well on measuring the REJ domain, which stands for receptor for egg jelly. So it's common that they've both been researched in the past together, the PKDREJ protein and the REJ domain, because it's found in the extracellular region, where a high concentration of sites under positive selection has been detected for sea urchins actually, and primates as well in previous studies. So 24 rodent species were included from the superfamily called Moridia. The gene sequences for rodent species were obtained using amplification using PCR actually, so they did actually have to perform some extraction and experimentation for the rodent species. But to detect adaptive evolution, the phylogenetic tree was divided into foreground in the front and background branches, while only the foreground lineages could exhibit positive selection, while the background obviously just stayed the same. So you could see the differences between the two. Um, Relative testes mass was something that was used to measure the influence of postcopulatory sexual selection on reproductive traits. So they actually use testes mass relative to the species' actual body mass to measure how much sperm competition there was. It's something that I didn't ever realize that people actually, or that something that was possible at all until they cited some other studies previously, I think they were from 10 or 15 years ago, that basically prove that this is a good method for testing whether sperm competition was present. So they use the testes mass of both rodent and primate species and how they were changing relative to their own body mass. For rodents, body mass and testes mass were obtained in the laboratory. A total of 5 males for each of the species, of the different rodent species families, were sacrificed actually by cervical dislocation and weighed, and then the testes were removed and weighed as well. For primates, they didn't have to use those methods. They didn't sacrifice any of the primates or anything, but they didn't go over specifically exactly the methods they used for weighing the testes in the primates. You can see on the phylogenetic trees onto the left, they use two strategies. They use maximum likelihood and Bayesian, I'm not sure if I'm saying that completely correctly, Bayesian analysis. In the case of maximum likelihood analysis, statistical support for each internal branch was actually estimated using bootstrap of 100 replicates. So that's how you can see the numbers between it, kind of showing basically the evolutionary distance between each of the species. And you could see on the bottom of each one, both of the methods had an outgroup for, I can't really see exactly, lemur cata. I'm not sure if I'm pronouncing that correctly. I think it's Latin, but. So the researchers used regression analyses to examine the link between PKDREJ divergence, or its evolution, and levels of sperm competition in primates. And no significant associations between PKDREJ divergence or evolution and testes mass, or for the extracellular REJ domain that we mentioned previously, were detected. So there was no correlation, meaning that it wasn't being selected for, or positively selected for. So as you can see again on the right, this one's on the right doesn't show any of the primate species. The one on the previous slide is actually the one that showed the primate species. These two on the right show the two rodent families that were used from the superfamily that we mentioned earlier. But the construction of these trees of the PKDREJ phylogenies were done by maximum likelihood and Bayesian methods, but they produced the same topologies, as you can kind of see from the right, very similar to the last ones, but just done with the species from the rodent group. But in rodents, specifically those in the family of Crickettidae, I'm not sure if I'm saying that right, there was a higher divergence of PKDREJ, specifically in the lineages that were detected to have high levels of sperm competition, which were obviously identified by the testes mass. So meaning those individuals that were detected to be partaking in sperm competition, meaning that there was one egg present and multiple sperm competing, those individuals did have a higher divergence of PKDREJ, meaning that it was evolving and increasing the likelihood that positive selection was occurring in those individuals. But that's only for the rodent species. And if you want to get very specific, positive selection is mainly concentrated on the two species mentioned in the bottom bullet point below, Cricketillus griseus. I'm not sure these are, I'm guessing Latin words. I'm not going to try to pronounce the other one, which were indeed, the researchers mentioned, indeed the species with the highest inferred levels of sperm competition, kind of inferring that the higher your level of sperm competition, measured again by testes mass, the higher divergence or evolution rate or positive selection of the PKDREJ gene. So at best, the results of this study reveal that PKDREJ sperm protein that appears to modulate the timing of fertilization through a role in sperm transport in the acromosome reaction shows bouts of adaptive evolution associated with the sperm competition we've seen. So just bouts of adaptive evolution, and again, that's not in the primates, just only seen in the rodents. So I think you could say overall it didn't exactly prove what the authors I think had written as a hypothesis. It did for the rodents in a way, not exactly as they described, but it did show, and it was the first study to show kind of what they were in the direction of what they were expecting to find through this research. And this is a specific resource, the only resource I use, and the study that was done, you can see